Okay, everyone, let's get started. First, I just want to thank everyone for coming out this morning to Workshop 29, Don't Stop Believing, Solutions to Read-Through of PTCs. This is mine and Margarita's disclosures. So we have a lot of good talks scheduled today. We tried to pick a couple different therapeutic avenues for read-through of CF-causing PTCs, including a genome-wide knockout screen to identify novel targets, suppressor tRNAs, small molecules such as the ERF3 degraders, and also some other small molecules that actually enhance other read-through agents, including the ERF3 degraders and aminoglycosides. So with that, we'll just go ahead and start with Eric Wong from the CFFT lab.
Good morning, and my name is Eric Wong. I'm a researcher at the Cystic Fibrosis Foundation Laboratory in Lexington, Massachusetts. And I would like to begin by thanking the organizers for the opportunity to share our work here today. And I have no disclosure. So, and roughly 10% of people with cystic fibrosis carry a nonsense mutation in the CFTR gene. So, and this mutation introduces a premature termination codon in CFTR mRNA, resulting in truncated CFTR protein and triggering CFTR mRNA degradation. This leads to absence of functional CFTR protein and severe CFTR phenotype. One therapeutic approach for nonsense mutation is the use of readthrough agents that suppress translation termination at PTC to restore synthesis of full-length CFTR protein. And aminoglycosides like G418 are among the most potent readthrough agents. But clinical trials have shown limited readthrough efficacy and toxicity with prolonged exposures. Currently, there's no therapy for people with for CF patients with nonsense mutation, highlighting the urgency to identify therapeutic targets that can enable pharmacological intervention to achieve meaningful readthrough efficacy. Translation termination factor, ER1 and ER3 have emerged as promising targets for enhancing PTC readthrough. Silencing ERF1 via siRNA, as shown on this Western blot, synergize G418 to increase full-length CFTR production. Combining readthrough agents and complementary treatment may improve readthrough efficacy and enable dose reduction to reduce aminoglycoside-associated toxicity. In this context, we envision a genome-wide CRISPR-Cas9 knockout screen in the presence of G418 to uncover genes influencing PTC readthrough as potential therapeutic target for readthrough therapy. We used the ER1 and ER3 as positive control in our screen. A gene is considered as a lead if it enhanced it at least 20% readthrough of ER1 benchmark in the context of native CFTR expression. So, to implement the screen, we first generated a doxycycline-inducible Cas9 stable cell line using our gene-edited 16-HBE CFTR G542X. Because local sequence content affect readthrough efficiency, we created a fluorescent-based readthrough system, including CFTR PTC sequence. This involved cloning a 55 CFTR amino acid peptide surrounding G542X upstream of GFP into a lentiviral expression vector. Transduction, the inducible Cas9 cell produced a CFTR GFP readthrough reporter line. Where successful readthrough would result in expression of a GFP fusion protein, allowing a fluorescent-based quantification of readthrough activity. Early A heat validation revealed that a heat-induced CFTR expression through a non-canonical initiation codons downstream of G542X PTC. This suggests that gene lockout promoting alternative initiation Readthrough could appear as a hit in our screen, independent of PTC Readthrough, leading to a false positive hit in the screen. To address this, we created a second Readthrough reporter line with a PTC in GFP coding region as GFP E112X. Additionally, we constructed a third Readthrough reporter line for high-throughput heat validation using native full-length CFTR R1162X. This system included a narrow-loop luciferase fused to CFTR C-terminus and an EF1 alpha promoter to boost transcript level. Final heat validation was performed in CFTR R1162X to assess heat in native CFTR genomic content. We chose a guide-in CRISPR-Cas9 genome-wide single-guide RNA library system from Takara Bio. This library is based on Berlando library, containing 76,000 single-guide RNA and targeting 19,000 genes. To monitor the guide representation, we performed next-generation sequencing and bioinformatic analysis at each key steps. Genes were considered as hit if their associated guide showed it more than 30-fold enrichment in a GFP-positive sorted cell compared to the unsorted population. For the screen, starting with the Takara Bio plasmid library, we generated a pooled lentiviral library and transduced the reporter cell at the MOI or 0.3 to ensure only single guide integration per cell. Following transduction, cells were selected by hygromycin and expansion and Cas9 induction. Then the cells were treated with G418. We collect the cells at each step for NGS analysis. Enriched guide were identified using our in-house bioinformatic pipeline. Applying a 30-fold enrichment cutoff, we identified 160 candidate hits, including our positive control EF1 and EF3A, validating our screen effectiveness to uncover regulator in the PTC Readthrough pathway. To eliminate false positive from alternative initiation, we used the GFP E112X cell line instead of the original screen cell line for the heat validation. Hits were ranked according to their full increase in fluorescence intensity compared to G418 treated control alone. Using a threshold twofold or higher, 77 hits that promote PTC Readthrough were validated in this system. We further evaluated the 160 hit using the EF1 alpha CFTR R1162X luciferase system. Only 4 hits out of the 160 hits showed it twofold or higher luciferase signal, indicating a potential to increase CFTR Readthrough in a more physiological relevant contact. Finally, we assessed the 4 confounded hit using a 16 HPE CFTR R1160 XL. Western blot analysis revealed that none of the confounded hit demonstrated any readthrough in this native CFTR contact. In summary, our genome-wide screen initially validated 77 hits that promote PTC readthrough in the PTC reporter contact. Secondary hit validation confirmed 4 hits in the boosted CFTR genomic content. Unfortunately, none of the confirmed hits demonstrated any readthrough activity in a native CFTR genomic contact. Therefore, alternative approach for future screen needed to achieve, in order to achieve pharmaceutical meaningful level of PTC readthrough, need to be screening directly in the native CFTR genomic contact, or focusing on combinatorial approaches that harness additivity or synergize between 2 known mechanisms, translation termination and MMD inhibition. And I would like to thank all my colleagues, particularly the NGS and bioinformatics team, for their contribution to this project. Thank you. Thank you.
Is this on? Okay. So one of the limitations for doing the CRISPR knockout is that you are dependent on loss of function based mutations. I was wondering if you are interested in doing more of a random mutagenesis where you could get, a gain of function type of mutation that would allow you to get read through, or even something that would be more specific and not so, breaking the entire machine of read through specificity.
But this is a single guy, so it's supposed to only manipulated one particular gene in a single cell.
Yeah. I mean, but you're still limited to whether or not that protein itself isn't functioning at all, right? Where instead you could end up maybe making it better at, right, with a gain of function type approach for the screen.
Yeah, I mean, there's a different approach. Instead, like the logout, you can always just screen the interference, and then another way just those SI and silence. Yeah, this kind of approach. Yeah.
Okay.
So following up on that, so if you hit one of the 2 alleles, you're only going to reduce it by 50%, right?
Yeah.
Right?
Possible.
Sometimes, unless you actually get both alleles. And so did you look at how efficiently you were getting a double hit versus a single hit?
We have tried to understand the screen system at the very beginning after we have the first set of the hit list. We selected the top hit, middle, and few of nothing to do with the readthrough. And then we test all of the 4 individual guys and test them individually. We are able to confirm 27 out of 28 of the guy have sufficiency lockout of the target based on Western blot.
But both targets?
I don't know both of them yet. So we only know. Yeah.
Do you know if you lost complexity in the library? If something was lethal, a knockout is completely lethal, but it might be ameliorated by a small molecule down to a survivable amount?
Yeah, so I don't know, but because the guy representation, the conservation is so important for this like the genome screen. We check like the guide representation and every single step, and then see here, potentially, of course, because this one is almost like a 2 weeks assay, if any essential gene could be dropped it out in this process. Yeah.
So anyway, the point I was trying to make, if you hit both alleles of either ERF1 or ERF3, it's lethal because they're essential genes, right?
Yeah, but we don't know because, like we just simply cannot check all of them. Yeah.
So when you did, I really enjoyed that. When, what was your library coverage? So how many cells did you actually transduce? So if you did like one allele, you probably would have a coverage of 10-fold, 100-fold.
Yeah, so it's 1,000.
1,000. There you
so
go.
basically, by default, it's like for the same guy.
Yeah.
Should have 1,000 gene carrier. No, 1,000 cell carrier.
So that would take care of the question if you've hit one or two alleles, because if you have 1,000-fold, and in those thousands, you're going to hit 1, 2 alleles. Yeah.
So, it's also, we are doing more than that. For this one, you only need about 50 million cells. We screen more than 100 million. Yeah. And also there are 4 guys per target, so you can multiple. Yeah.
Have you considered using a different readout?
Such as
fluorescent labels?
Yeah, so this one is GFP.
Yeah, but not related to the processes themselves, related to the read-through at the NMD. Yesterday I showed what we are using for a drug screen.
Yeah.
And so it's a double-labeled, a triple-labeled CFTR mini gene, which actually has M-cherry at the N-terminus, GFP at the C-terminus, so we can have different readouts for NMD inhibition or read-through activity.
Yeah, I think that is a good suggestion. Are we pointing to Martin?
I was just going to briefly chime in. Yes, there are other systems, and you have done nice work with it, Margarita. I think one of the big points here was, Eric had hits until we went to the most stringent native system, right? So basically saying that all the gene candidates that were identified in more permissive screens ultimately didn't have enough efficacy to still meet the criteria that we were happy with it in the most stringent system. So I don't think using...
Based on knocking out those genes, or what was the criteria for the failure? So
they were knocked out in the original screen, but confirmation was also at some level done with shRNA or siRNA. So we allowed for hypermorphic kind of mutations. Ultimately, as I say, nothing of that really stayed above where we thought it was meaningful for us to pursue further.
Okay, we can rescreen those 160 genes if you want.
Sure. Yeah, but thank you, Martin. I would say this under our screen condition.
Can I ask one question here?
Sure.
Yeah. So your data is compelling in a way that you ended up having none of the targets for this. So for any big screen that you have to do, I think a compelling experiment would be, what is your positive control? Here, your positive control is G418. But in CF, what we have learned, it's a genetic disease and with a lot of heterogeneity among the variants. I would suggest one example for you, that if you have to take a positive control in terms of the variants to find, it has to be something which responds to these modulators. And one of the best examples I could suggest to you that has been tested in primary cells are the N-terminal nonsense variants. Individual with R75X who is homozygous, we found that it responds beautifully to ELOX. ELOX has failed in the clinical trials, and the reason being, again, because they were going with the numbers. Numbers of G542X, which is very common. R75X homozygous has been found in an individual with Indian descent, very rare variant. But nature has allowed these variants to use N-terminal alternative methionine, which leads to RNA stability. My only suggestion for you when you are doing such a big screen is, please include one variant that is known to respond to read-throughs in the primary cell settings so that you can set the stage. We have learned from G551D, which was only 1.25% of the CF population responded to ivacaftor, and that is how you have expanded today to F508del. So please try one variant which you find has responded in the primary cell setting.
So, and indeed, G4 is not our positive control. This is our screen background. Our positive control ER1 and ER3A. So you work on all the cells we have been tested. Yeah.
And it works in primary cells.
Yeah, primary cell. Yeah.
So one last thing, people in yeast have done screens and screens and screens, and there's a lot of these weak suppressors of nonsense, and that's probably what you're getting. So you're not getting the big ones like ERF1 and ERF3 because they're lethal.
Yeah.
But if you looked at your hits, you're probably reproducing what they saw in yeast. That would be my guess.
Yeah, I would just emphasize this is only one screen condition. So maybe, you screen under different condition, you may able to achieve the goal. Okay.
Yeah, just one clarification on the library construction. So this is predominantly protein coding genes that you're targeting.
Yes, yes.
So you're not hitting any like snow RNAs, which could potentially be creating suppressor tRNAs and stuff.
So at the time when we choose this library, we believe this is the best library for this type of the work. Yeah.
Okay.
Thank you very much.
Thank you.
So we proceed to our next speaker, Yunji Wang, if I got it correctly, and coming from Indiana University, and the title is CFTR glutamic acid nonsense mutations are rescued by a newly engineered suppressor transfer RNA in a variant-dependent manner.
Thank you. Thank you, everyone. My name is Yunji Huang, and from Indiana. First, I would like to thank the organizer for the opportunity to share my new project. It's really new. I worked on this project about a little bit over a year, which focus on developing suppressed tRNA to restore CFTR, which is affected by premature termination codon. Specifically, I'm going to talk to you about a newly engineered glutamic acid tRNA. There's nothing to disclose for this presentation. And we all know that CFTR is a chloride channel expressed on the plasma membrane. And there's a lot of mutations. And based on the effect of this mutation on CFTR protein, they can be grouped into different classes. Specifically for the class 1, they are nonsense mutation. They are not eligible for CFTR modulator. And currently, there's no really FDA-approved drug to treat patients with CF carrying this class mutation. So mutations in this class are largely premature termination codon, such as G542X, W1282X, and R553X. So what actually premature termination codon? So they are essentially single nucleotide mutation cause sense codon to mutate into a stop codon, such as UAA, UAG, and UGA. As a result, the affected mRNA transcript failed to translate it into a full-length protein. And currently, there are several technologies under development to treat diseases caused by PTC, including translational read-through inducing drug, antisense oligonucleotides, and CRISPR-based gene editing, and the one I'm going to talk to you, which is suppressed tRNA. So what is suppressed tRNA? They are essentially nature tRNA, which has been engineered. The engineering process usually involves mutating at least the anticodon part of the transfer RNA to enable complementary base pairing with the PTC and enable the incorporation of amino acid at the PTC position, restoring full-length protein synthesis. So this concept is not really new. Actually, it was developed almost half century ago. And this is data from a very early study using suppressed tRNA as a therapeutic approach. And they investigate the case of beta thalassemia, in which beta globin protein was affected by PTC. And it's an in vitro translational study. And essentially, the study revealed that in the presence of ACE tRNA, additional signal emerged which matched the beta globin protein, suggesting suppressed tRNA able to suppress PTC and facilitate protein synthesis. Recently, there are many groups regained interest in this technology, and here just a list of publications in a high-impact journal really indicate the suppressed young is regaining significant attention as a therapeutic tools. And just give you a taste of what they have done, and this is one of the study in that publications. And in this mouse model, the hyaluronidase gene was affected by PTC, and they inject the mouse with recombinant AAV expressing suppressed tRNA, and this AAV suppressed tRNA able to restore the hyaluronidase activity in both liver and heart as well as other organs as well. So suggesting the in vivo application of suppressed tRNA is possible to treat genes containing PTC. But not all the ACE tRNA is effective. This is what's done by John Luke's group. He's in the audience. So compared to other ACE tRNA, and glutamine acid ACE tRNA is one of the least efficient. And you can see they basically essentially generate all the ACE tRNA and compare their suppression efficiency. The x-axis is the log scale in the suppression efficiency, and the blue line is the 100-fold correction mark. And you can see, glutamic acid ACE tRNA not able to surpass that line. So according to CFTR2 database, glutamic acid PTC is really mutation hotspot, representing roughly 8% of CFTR PTC, and notably E60X is the 6th most frequent PTC. So it's worth it to improve this ACE tRNA. And here is another example just show you ACE tRNA for glutamic acid is not as efficient as others. So the question for this project is, can we further engineer this ACE tRNA to increase its efficiency? So how are we going to do that? So we really need first to establish a system that allows us to measure the suppression efficiency. So we designed this plasmid, and which contained one copy of ACE tRNA and a test gene. In this test gene include Ranella luciferase, test gene containing PTC, and firefly luciferase. And in this condition, Ranella luciferase expression is not affected by PTC, but firefly luciferase expression depends on PTC. So we use a ratio of firefly luciferase to Ranella luciferase as an indication of the suppression efficiency. And we transfect this plasmid into H1299 cells. So we select two ACE tRNA, natural tRNA for glutamic acid, 1-1 and 1-2. And for optimization, we began by engineering the anticodon only to generate ACE tRNA we just mentioned a couple slides ago. And consistent with previous study, these two ACE tRNA has really low suppression efficiency compared to the control, and they are not even significantly higher than the control tRNA, which cannot be charged by any human amino acid or tRNA synthetase. Next, we sought to identify if there's any mutation we can make on this tRNA to improve PTC read-through efficiency. So we specifically focus on the anticodon loop and anticodon stem, believing that, fine-tuning of this nucleotide, we're able to compensate the change that at an anticodon. So we begin with this A38 mutation, and we found that this A38 mutation able to improve suppressed tRNA 101 significantly, around 24 increase, but has nothing to do, no effect on the 2-1 tRNA. And interestingly, when we mutate another one for the 2-1 at the 32 position, and we see the improvement in the suppressed tRNA efficiency. Furthermore, we explore the mutation to the anticodon stem. Anticodon stem is important to stabilize the stem helical conformation. And we found that introducing this A31U39 paired to the 1-1 tRNA actually decreased the suppression efficiency, but marginally increased this 2-1 tRNA. Lastly, we found that additional G30C40 mutation to 1-1 tRNA further improved this tRNA efficiency. So what we learned from this study is that the tRNA efficiency can be improved by modulating the anticodon loop and anticodon stem, but also the different tRNA probably require specific mutation to reach their optimal conditions. But these studies were not able to demonstrate whether the installed amino acid is glutamic acid or not. And then we designate this highly effective suppressed tRNA for glutamic acid as V13 as for our following study. So to identify the amino acid installed by V13 at the PTC encoded position, we utilize a GLP protein reporter gene, which had a UAG codon at 150 and a strapped everything actually at the C-terminus for the purification purpose. The plasmid, again, is transfected into the H1299 cells. Clearly, this V13 able to restore GLP protein, confirmed by fluorescent microscopy and as well the Western blotting. And also, we observed the suppression efficiency comparable with our previous screening experiment. And we purified this GLP protein and analyzed them by tandem mass spectrometry. We found that 90% of the protein installed at glutamic acid position is glutamic acid. So demonstrating V13 predominantly installed glutamic acid at UAG codon. Next, we investigate whether optimizing V13 expression could lead to further improvement in PTC suppression. We first explore the effect of using different promoter to drive tRNA transcription, including 3 common promoters, 7SK, H1, and U6, as well as one synthetic promoter, M11. In this experiment, a plasmid containing the secreted embryonic alkaline phosphatase as the promoter, it has a UAG stop codon at the 40 position. And all promoters clearly exhibit very similar level of PTC suppression, demonstrating very minimal impact on the choice of tRNA promoter. Next, we ask whether we can increase the copy number of the tRNA cassette to improve tRNA suppression efficiency. It was found that actually, 3 copies of suppressed tRNA had over 2-fold higher UAG suppression than the plasmid with only 1 copy, suggesting increasing the number of suppressed tRNA can substantially enhance suppressed tRNA efficiency. So with this study, we want to investigate whether V13 could suppress PTC in CFTR gene. We select 2 CF-causing glutamic acid PTC, E60X and E1418X. We co-transfect this plasmid in HK293 cells, 2 plasmid, CFTR containing PTC, and then these 3 copies of tRNA plasmid, either V13 and the control. Western blotting analysis show that V13, but not the control tRNA, able to significantly increase the amount of mature CFTR for both of these mutations. And we also include delta L508 treated with CFTR modulator for comparison. And data suggests that V13 tRNA able to restore comparable level of mature CFTR to L508 delta CFTR treated with CFTR modulator. Suggests this tRNA may be able to achieve the therapeutic efficiency. So next, we want to test the functionality of this V30 rescued CFTR. This was done by fluorescent-based SPQ assay, and in both with E60X and E1418X, we noticed a significant increase of CFTR function rescued by V13 compared to a control. And also, we have the alpha weight, delta alpha weight, with treated with CFTR modulator. So collectively, these data suggest V13 tRNA is effective in rescuing pathogenic PTC and leading to restoration of functional CFTR proteins. And lastly, for this project, we want to know if V13 can equally restore all glutamic acid PTC in CFTR. To do that, we generate additional two glutamic acid PTC, E664X and E831X in CFTR for comparison. Both are located in the regulatory domain and of the CFTR protein. And interestingly, V13 able to improve E831X CFTR stronger than the E664X, suggesting they are V13 rescue CFTR PTC differently. To further support this observation, we turn to another tRNA, arginine-based tRNA, and to target different arginine PTC in CFTR. And consistently, the response of these arginine PTC to ACE tRNA varies. With R75X, it's been the most efficient, but the R553X and R1158 be the least efficient. Collectively, these data suggest suppressed tRNA is unlikely to rescue each PTC equally. So what we learned so far, we learned that modifying the ACE tRNA nucleotide has the potential to improve the efficiency of ACE tRNA, and each tRNA probably requires specific modifications to reach its optimal form as a suppressed tRNA. And also increasing the copies of suppressed tRNA can lead to improved PTC readthrough, and each PTC mutation probably need a specific suppressed tRNA for optimal rescue. With that, I would like to thank people who work with me and also the collaborator, the group people for their continuous support, and also definitely want to thank the funding agent, especially CL Foundation, for continuous support. And with that, thank you for your patience.
Very nice presentation. Perhaps I can start with questions. So you actually changed or optimized your tRNA at the anticodon and stem, because that's for obvious reasons to increase the read-through ability. Have you also considered changing the tRNA to improve the affinity for amino-acyl tRNA synthetases? Because in the end, it will be competing with the native tRNAs.
Yeah, that's a good question. So yeah, we try actually, I didn't mention in this presentation, we changed that nucleotide, which we think is important for affinity to the elongation factor. And yeah, we also did other nucleotide as well, but we don't see further improvement. Yeah, this is the best one, yeah, so far.
Okay, but doing these enzymatic competition studies with the cognate tRNAs might be helpful in terms of giving a preference for the amino-acyl tRNA synthetases.
Okay, thanks. Thanks for coming.
John? Oh, sorry, Gary.
I probably missed a nice presentation. I probably missed this. The target gene, was it a cDNA or was it the actual gene itself?
Yeah, cDNA. But we are now testing in the primary cells. Yeah.
Okay. So when you test it, I think the two that you're looking at probably have stable RNA, the two variants, right? So 5' ones, as Niraj mentioned before, the alternative methylation met site in CFTR permits stripping of the EJC proteins, which stabilizes the transcript. So anything that's, 5' that Margarita's worked on this and shows the protein's partially stable and everything else. So consider that not all these nonsenses are working the same way. Yeah. And the ones at the 3' end also tend to be relatively stable as well. So I think you picked two really good ones to work on to demonstrate it, but doing it in the gene would be really helpful. You're considering the transcript as well. The other question I had, sort of related to what, if you don't mind me asking that question.
Sure.
The other concern you have with modifying these tRNAs, and you get them more and more effective What happens to other sites, unwanted sites, if they're getting more effective? Are they going to finally interfere with native termination codons? Are they going to allow the expression of unwanted proteins? So I'm always worried about specificity as well as effectiveness. Can you use the +4 nucleotide? Because that is part of the reading system for the ribosome. So can you use that to help specify not only the 3 nucleotides, but the fourth, or even ones that are on the 5' end of it to increase your specificity as you do your screens? And that way, maybe it'd be more specific for that particular termination codon. All right, I've taken more of my time. Thank you.
Thank you. It's very creative idea. Yeah, we didn't test that. I attended a couple of workshop yesterday, and actually in the translational process, ribosome actually recognize 4, not just 3. So I think that your idea is it's definitely worthy to test. Yeah, thanks. Yeah.
John?
That was a beautiful talk. So, you made modifications to the T-stem and the anti-codon loop and kind of like insider tRNA based, baseball. But have you looked at the processing? So the 5' and 3' of the tRNA that's important for it to be cleaved by RNase A and P. Did you make modifications in those regions as well, or swap out to see if you can improve?
Great question. I noticed your publication, you're looking at that area. I think it's worthy to do that. But right now we just focus on can we develop the most effective suppressed tRNA or ACE tRNA to target these PTC. But that's definitely is a future direction. Yeah.
And to Margarita's question about the amino-acyl tRNA synthetase, we've done experiments where we've overexpressed that synthetase to see if we can improve charging and suppression efficiency. And indeed, you can with some of the tRNAs, but not with all. So that interaction likely is very limiting, not just in suppressor tRNAs, but in all tRNAs. Everything is different, right? So
yeah.
Thanks.
I have one more question. Okay. So I couldn't really see it very well, but it sounded like you said only 90% of the amino acid that was incorporated was 95.
Yeah.
So what was the other 5, and how do you propose that it became incorporated? Like, was it mischarged, or was it like a near cognate? Like, what was the other 5?
I think it's probably, I don't know the answer. That's a quick question, quick answer, but it could be lead by mischarge, very likely, I would say. Yeah. We tested on the charging process actually in vitro, but it's in vivo, in cells condition, we don't know. So yeah.
Okay. Thank you.
Thank you very much.
Oh, thank you.
Okay. Up next, we have Lisa Nicholson, who is a PhD candidate from Case Western Reserve. And the title of her talk is Targeted ERF3A Degradation Amplifies Nonsense Mutation Read-Through in a CF Murine Model.
Hello, everyone. I'd like to thank the organizers for inviting me to speak today, and thank you all for attending my talk today. I'm excited to share with you my project investigating ERF3A degradation as a potential therapeutic treatment for cystic fibrosis nonsense mutations. I have nothing to disclose. So as the 2 speakers ahead of me already mentioned, since the identification of cystic fibrosis, there's been large advancements that have been made in the development of a cure and symptom management, as today over 90% of people with cystic fibrosis have mutations that effectively respond to the CFTR modulators. A cure is still needed, though, for the remaining individuals who have 2 rare or complex mutations, including those with class 1 nonsense mutations. Nonsense mutations result in premature termination codons, which lead to mRNA transcript destability, and if protein is produced, it is often truncated and non-functional. An effective nonsense mutation therapy will need to both increase the mRNA transcript abundance and induce read-through of the PTC to produce functional protein, but current treatments that are effective in cell models to induce read-through or inhibit NMD have toxic side effects which prevent their use clinically, or they produce very limited positive effects, meaning an effective treatment is still needed today. Targeted degradation of ERF3A is one method of nonsense mutation therapy that has shown promising results when co-treated with a read-through agent. ERF3A is a protein crucial in stop codon recognition as it binds to ERF1 and interacts with the ribosome at a stop codon to trigger peptide release. Inducing partial degradation of ERF3A may allow more time for a near-cognate tRNA that matches 2 of the 3 nucleotides of the stop codon to bind to the PTC and induce read-through and improve the mRNA's stability. ERF3A degradation is achieved through cereblon modulators, which modify the substrate specificity of the E3 ubiquitin ligase complex displayed here. The 2 compounds, CC9009 and SJ6986, or SJ for short, both induce ERF3A specific degradation. These compounds have thalidomide-like binding motifs, allowing them to bind cereblon, but unlike thalidomide and its derivatives, CC9009 and SJ are specific to ERF3A and don't affect SAL4 and the other proteins that induce the negative side effects thalidomide is known for. Now, despite the birth effects that are associated with thalidomide, it is prescribed clinically today and used for its anti-cancer and anti-inflammatory properties. This might indicate that the cereblon modulators may be clinically tolerable, especially since their targets are not associated with the same toxic side effects thalidomide and its derivatives are. The cereblon modulators have already been shown to amplify read-through of PTCs. A paper here from Krabi et al. Using the breast sarcoma cell line showed via Western blot analysis with the vinculin loading control that the co-treatment of CC9009 with the read-through agent G4 and 8 induced production of the full-length protein from the TP53 gene containing a nonsense mutation. This protein restoration was only seen minimally when CC9009 was treated alone and was completely absent in the G4 and 8 only treatment. Additionally, the SJ compound has also been used in cell lines created from a nasal curettage W1282X homozygous donor cells. Li et al. Showed in this paper that the SJ compound increases CFTR function as measured both by the Forsgren stimulation and CFTR inhibition, and co-treatment with G4 and 8 further increase the CFTR function restored. While there are papers such as these that administer the cereblon modulators to human cells and cell lines, the use of these compounds in murine models has been limited, as mice do not respond to these drugs due to species differences in the cereblon gene, which has limited the functional correction assays these compounds can be used for. 2 cereblon humanization mutations have been identified, though, and have been shown that they are sufficient for these compounds to degrade ERF3A in mice. We made a mouse with homozygous mutations and crossed them with the G542X CF mouse to allow us to use these compounds in mouse intestinal organoids and nasal epithelium to evaluate CFTR functional correction. The data I'll share with you today is using primary cells from these homozygous mice, and we will be followed up with in vivo treatments. Now, to start off, we want to confirm that the cereblon modulators do in fact degrade ERF3A in our mouse models by using mouse intestinal organoids derived from mouse small intestinal crypts and analyzing them through Western blot analysis. As you can see, both cereblon modulators degraded ERF3A in a concentration-dependent manner, and the concentrations used in the following CFTR functional assays decreased ERF3A expression to around 12% when compared to the untreated controls and normalized to EC adherence. To then assess the cereblon modulator's ability to restore CFTR function, the mouse intestinal organoids were then used for the Forsgren-induced swelling assay. For this assay, forsgren is introduced to the cell culture, which causes fluid uptake into the organoids, and the rate that the organoids swell is proportional to their amount of CFTR function. The area of all the organoids is measured every 5 minutes for 3 hours after forsgren is introduced, and then normalized to the time 0 area value. This is then graphed in a percent area increase normalized versus time for each well. Now, to give you a quick example of what this graph looks like, you can see here that the percent area increase for the wild-type organoids having normal CFTR function is displayed in black. The outlined circles are the G542X untreated organoids with no CFTR function, and the blue squares represent the G542X organoids after treatment. The area under the curve for the first 30 minutes is then calculated and used for the statistical comparisons between treatments, which I'll display in the further slides. Now, to measure the cereblon modulator's efficiency at restoring CFTR function, intestinal organoids were harvested from the mice homozygous for the 2 cereblon mutations and with the G542X mutation, and then treated for 24 hours with the read-through agent G418, the NMD inhibitor SMIG1I, the CFTR modulators, and the cereblon modulators in various combinations. So this is the area under the curve graph, which shows the percent area increase versus time for the first 30 minutes of the forced gland-induced swelling assay, and allows the CFTR functional correction comparison between the different treatments. The wild-type CFTR function is displayed here in black, and the previous in vitro best treatment is displayed in blue for comparison to the other cereblon treatments. So what did we find? We found that the cereblon modulator SJ6986 does not increase CFTR function on its own or when combined with the CFTR modulators. The combination of SJ with the G4 and 8 read-through agent produced some CFTR function, and the addition of the CFTR modulators further increased that function, showing that the combination of G4 and 8 and the CFTR modulators with either SMUG1I or SJ produce the same amount of CFTR function. We then finally combined all the compounds used and saw a further increase in the CFTR functional correction. I repeated this with the CC9009 drug and saw very similar robust CFTR function restored as per the SJ compound. Again, CC9009 on its own or with the CFTR modulators produced no CFTR function. Noticeably, though, the combination treatment of CC9009 with G4 and 8 and the CFTR modulators achieved higher CFTR function compared to the previous in vitro best in blue, which was not achieved with the SJ drug. Lastly, the addition of the SMIG1I to the G4 and 8 CFTR modulator and CC9009 treatment did not achieve any increased CFTR functional output, indicating that CC9009 may be more potent than SJ6986 at amplifying read-through. After establishing that the cereblon modulators produced function in the intestinal organoids, we then wanted to use real-time PCR to measure the abundance of the CFTR transcripts to determine if they stabilize the CFTR mRNA at all. Now, the nonsense-mediated decay inhibitor SMUG1I increases the CFTR expression to around 75% of wild type when treated alone and to nearly 90% when treated with the G4 and 8 read-through agent. CC9009 on its own achieved minimal restoration, but when co-treated with G4 and 8, the CFTR mRNA expression increased to nearly 50% of wild type, and similar results were seen with the SJ compound. This increase in the CFTR transcript abundance by the cereblon modulators may be because they are inducing very effective read-through of the PTC, thereby removing the exon-exon junctions that are downstream of the PTC and preventing NMD targeting, though direct NMD inhibition by SMIG1 is more effective at restoring the expression levels. We then wanted to confirm that results we were observing with the read-through agent G4 and 8 were repeatable using a less toxic read-through agent. While G4 and 8 is used in vitro, it's not clinically prescribed due to toxicity, but the alternative drug ELX02 has been used in clinical trials before and is safer, though it is a less potent read-through agent. Now, excitingly, the area under the curve results from the initial tests show that ELX02 achieves very similar CFTR functional restoration as G4 and 8 when co-treated with the cereblon modulators. And this might indicate that the read-through agent ELX02, despite its unimpressive clinical trial history, may be needed to be combined with other compounds like these cereblon modulators to be truly effective for nonsense mutations. And then lastly, we used the mouse nasal epithelium cells to repeat the treatments in a different primary cell type. To measure CFTR function in mouse nasal epithelium, the mice are sacrificed, the nasal epithelium isolated, and then grown on a filter membrane. Using the air-liquid interface cell culture conditions, the epithelium is differentiated and can be used in an oozing chamber to assess CFTR function. The figure on the left here shows the raw data obtained from one mouse nasal epithelium experiment. Amiloride is added to inhibit sodium channels, forskolin is added to activate CFTR, and then the CFTR inhibitor allows CFTR function to be measured from the change in current. Excitingly, the cereblon modulators produced robust CFTR function in the mouse nasal epithelium, as well as what was seen in the intestinal oosings, or the intestinal organoids. Notably, 33% of the wild-type current was achieved by CC9009 treatment, and around 28% of the wild-type current was achieved by the SJ treatment. This work shows that the cereblon modulator response was not cell type specific, and this supports the use of these compounds for our next steps in research, which is in vivo use. So in summary, today I've shown you that the cells from the G542X mice humanized for cereblon react to the CC9009 and SJ6986 cereblon modulators. The cereblon modulators enhance the read-through agent efficiency in both the intestinal organoids and the mouse nasal epithelium, and the cereblon modulators increase the transcript stability of CFTR when co-treated with G4 Lastly, moving on, I think our results are reasonable for us to continue this research in our in vivo use. So for our future directions, we plan to administer the cereblon modulators in vivo and confirm ERF3A degradation and determine the best dose to use. After that, we will administer the cereblon modulators with the CFTR modulators and ELXO2 in vivo and measure CFTR functional correction. I'd also like to perform some ribosome profiling to calculate the rate of read-through achieved and confirm how the endogenous stop codons are being affected. And then lastly, I'd like to look more into how the cereblon modulators are affecting the ENaC channel, as research out of the University of North Carolina from the Scott Rendell lab has shown that ENaC inhibition has been observed after cereblon modulator treatment, which is a potential to be an additional therapeutic benefit for CF treatments. And with that, I'd like to thank everyone involved with the project, as well as our funding sources. Thanks.
That was very nicely presented and very interesting. Any evidence of toxicity? Did you get any hints from the mouse cell studies or the in vivo studies?
So I haven't measured toxicity directly besides just looking at the organoids if they look healthy or not. At the doses that I was using for the analysis here, there doesn't seem to be much toxicity. They don't look as healthy as untreated, but compared to the SMUG1 treatments, they definitely look more healthy.
That really great job. Thank you for sharing that. So the future studies going in vivo, you know, the mice that are insensitive to thalidomide, they were a great model for treating with these drugs, but now they're going to be sensitive. So I wonder, like, how much trouble your IRB is, IACUC is going to give you about treating these mice. What are the kinds of studies that you have to do up front to understand the dosing and that kind of thing?
So we've already been approved to use these compounds in mice, which is great. But as I said, like our first studies are going to do different dosage, different durations of the cereblon modulators in the mice, just to see if we can detect ERF3 degradation and assess for toxicity that we see with the mice.
Yeah. And one option that might be useful, because of course, when we've seen this drug work and we've been wondering about could it ever be used in people, would topical administration, for example, to the nose, you know, prevent any systemic toxicity? So that might be a good option to consider.
Yeah, the method of treatment will definitely be important to consider, as well as when in development is when you can start administering the drug. We're going to start planning on treating mice that are around 8 weeks old with the drugs. So I'm not sure about in utero treatment with these. That's still that's a long ways out from there. But yeah, there's definitely certain things we'll have to keep in mind when treating the mice.
1 point here. So the question that Scott raised, I think that is more important, that has nothing to do with CF, about the IACUC. And why this point intrigued me is because next week I'm going to give a lecture to the students on animal ethics research. So for you, if you're a graduate student or a postdoc, I would say that always the answer comes up with refinement, reduction, and replacement. And what is the societal benefit of using these animals? Regardless of all this, I think the replacement comes very handy, And what you are doing along with Craig Hodges is so compelling that you are using organoids. So keep that always in your pocket, that if something happens to your animals, you are very well in that realm of using organoids and bringing this to the translational use, because you could use this in humans as well. And also look into BCAM and Degrasia's 6 Rs that they have come up with in 2020. BCAM, for some reason, he passed away this year, but please come and talk to me, because this will inform me as well. All right, thank you.
Thank you.
So if no further questions, thank you very much, Lisa.
Next, we have Corinne Augelli-Szafran from the University of Alabama at Birmingham. I'll put this here.
Yeah, sure.
Well, hello, everyone. It's nice to see a lot of familiar faces in the audience. This is my disclaimer. And, oh, boy. And my disclosure. I don't know what happened here. So I'm going to take a different avenue now. We're going to switch gears. You've heard a lot of great biology so far. My purpose here is to show how to move forward compounds, whether it's through medicinal chemistry and then, of course, the drug discovery effort. So I work a lot with Stephen Rowe, Dave Bedwell, the CFF Foundation, the CFFT Foundation, and it really is a great partnership to try to move these molecules forward using all of the extensive biology that's been presented today. So today, though, we have a lot of different classes of compounds that we're moving forward, and today I'm going to give an overview of some of the work that we did with the aminoglycoside enhancers. So this is probably trivial to many of you, but I'll review it anyway. Among the mutations which cause CF, cystic fibrosis, nonsense mutations result in a premature termination codon, PTC, which terminates the translation of the CFTR mRNA prior to generating a complete protein. So far there are no approved therapies that treat nonsense mutations that affect about 11% of the people with CF. Aminoglycosides are known to bind to mammalian ribosomes and reduce the fidelity of translation at the sites of PTCs, allowing insertion of a near-cognate tRNA. This process, we know very well as read-through, restores the translation after the PTC results in a full-length CFTR protein. So aminoglycosides, such as G418, which we heard a lot about already, have shown to promote CFTR read-through, but this class of drugs, as we know, is toxic for chronic administration at high doses, and from a medchem perspective, it's pretty huge and has a lot of labile groups on it. So our goal is to find compounds that enhance the read-through activity of aminoglycosides and ultimately identify a preclinical candidate that has clinical relevant synergistic activity. So the work I'll talk to you today about for these enhancers was the molecules that we chose to work on was found via a high throughput screen of a collection of about 500 compounds, and it was performed using 16 HBE cells with an analog reporter bearing the C542X CFTR context in the presence of low levels of G418. All the compounds were tested in the high throughput assays with and without G418. We did not identify for this particular screen any compounds that were active without G418. The effects on the CFTR function with selected hits were evaluated in 16 HBE CFTR G542X cell line using the TEC assays. Initially, we pursued 9 chemical series, and we identified 6 of those series with an EC50 plus G418 that hovered around EC50 of 1 micromolar. The other 3 series had higher EC50, so we put those aside. We looked at ADME properties, which is looking at metabolic stability, solubility, et cetera, to try to prioritize, since we had so many different chemical scaffolds to review, of which ones we would pursue from a medchem perspective. And shown on the right-hand side here is just really a pretty standard triage from a high-throughput screening perspective, where you use your SAR driving assays to select compounds, look at clusters of structural similarities, rank hits by their data, and then you choose your series to do the hit-to-lead optimization. This is probably a little bit too much data, but I just want to give you a sense of what we're up against when we're moving small molecules forward to hopefully preclinical trials. On the left-hand side, you'll see all of the different assays, the biology activity assays, the ADME properties, PK, and then off-target liabilities. When we're moving compounds forward, the criteria aren't as stringent as ultimately what you want as far as a target product profile to move forward with. But these were some of the criteria that we used to help prioritize and select compounds to move forward into our different assays. So we chose 3 scaffolds to work on, and based on the high-throughput screening data. Showing here are the data from the NanoLook reporter assay, and we were targeting greater than 100% Emax plus G418. And then when we got the ADME properties on these selected hits that we would do chemistry on, shown here is the MLM is the mouse liver microsome, human liver microsome solubility log D, and the criteria, you usually want, ultimately greater than 30 minutes here, 60 minutes here. Solubility, you want greater than 10, and log D between 2 and 4. So what we found is for this series here, had reasonable, this series here, we had reasonable metabolic stability, terrible solubility, and then the other 2 series, we had a lot of work to do to improve the hot spots found on the different scaffold to improve the stability. So our hit-to-lead optimization, as I showed you, we have these 3 hits. This is the HTS data. We did a lot of SAR, as I'll explain in the next few slides, on each of these series, and the best that we can do with this series, the 44473 series, was keeping the Emax plus G418 right around the 200 range. We were able to improve the EC50 with G418. We also always tested for cytotoxicity in the HBE cells at, I think it was 100 micromolar. So we always tossed aside any compounds that were, showing cytotoxicity. And then the tech assay, our goal was really 100%, but this was close enough since that was the best that we can do to highlight. In our purple series that we call the 44622 series, these were 3 of the main compounds where we really got up to about 500% here for the Emax plus G418. The other ones were hovering around 200. Our EC50s were, getting below the 1 micromolar range, which is really exciting, but we had a ways to go yet. And then we looked at our functional assay, the tech assay, and this one was a little bit above 100%. So, struggling, as I'll point out, with probably the drug-like properties of the molecule. The cell permeability is really important, and biosolubility also feeds into that, as well as metabolic stability. So it's really a balance of all of those properties. And then for the 44594 series, Emax plus G418 was quite impressive. We got a little bit farther below in the one in the EC50, 700 nanomolar, and then we have the tech data hovering around 90%. So for this 44473 series, this is stop-go type of SAR profile that I'm sure many people have seen. Green means, we're hitting the criteria. They're guidelines, though, really. The yellow is okay, and then the red, of course, we don't like. So for this series, we did SAR in 3 different areas on the scaffold, made a little bit over 100 molecules, and we purchased a few as well. The problem with this series is we had a hard time improving the metabolic stability and as well as the solubility. For the purple series, the 44622 series, I highlighted 3 of these main hits that we identified, showing here our high throughput assay,
the nanolook, the tech, and then all of the drug-like properties that we looked at with permeability, as you can see, is really not where we need it to be. We did SAR as well on this series in 3 areas and made a little bit over 250 analogs. And we were impressed with the nanolook activity we were seeing here, as well as with the tech data. But where we struggled, again, is with the solubility. And, in all of the different therapeutic areas that I work in and all the different scaffolds, this is always one of the issues. And sometimes it's the hardest one to improve. Of course, there's methods you can look at for formulation, nanoparticles, lipo, whatever. And, we're not there quite yet till we get the efficacy that we need, but we always do, from a med chem perspective, struggle with solubility. This, I just want to show you an example. Of course, we get full PK on compounds, and this is one of the compounds we did for 45583 in the purple series. And, if you look at the exposure here, it's quite low, and the bioavailability is quite low. We want this up into the 2,000, 3,000, 4,000 range. So, and of course, that probably is due to the solubility, permeability in the molecules, and also probably affect how it affects the activity we see in the tech assay. This work is the 45987 showing activity with and without G418. It enhanced the G418 activity when expressed as percent of the G418 EC50 values. So here is without G418. Here's G418 alone, no compound, and then here is G418 with the 45987. You can see a big enhancement here on the percent G418 EC50, which was really impressive. We also looked at 45987 with G418 and ET, the correctors. And shown here, you will see G418 alone, EC10, and then you see G418 EC10 with the compound 45987. You see an increase. Oops. And then shown here is when you add the correctors with G418 is shown here, and then when you add the compound, you see a big improvement here. So combining the 45987 with the G418 EC10, it provided a strong increase in the CFTR function compared to the same dose of G418 alone. So for the purple series, the 44622 series, in summary, the 2 of the main compounds that we explored at most, with PK, et cetera, had high nanolook and tech triple combo activity, had decent microsomal stability, poor solubility, was not toxic in the cells that we looked at. We also did more extensive studies that I won't go into, but 2 of them I'll highlight is the off-target profiling. So we look at a safety screen of 444 different enzymes or different receptors, and you look at a certain percent activity at a concentration, and there was no major activity for this series, which was good. The PK, as I showed you, we have to improve upon the drug-like properties of the molecule. I do want to mention that, these were the modifications for these 2 types of molecules, whether it was on a phenyl ring or one of the heterocyclic rings. But we identified, we modified the middle ring of this compound, and I was talking to Steve a little bit earlier today, that the solubility improved significantly. And so, we're carrying forward a lot of different series and for a lot of different purposes, but there is some work to do. I think with taking this change that we made, it had good permeability. Then now we need to bring in the activity and the efficacy in the tech assay. And, that's always a challenge when designing a molecule that has the right balance of properties. And we found another type of modification that we made in this series as well. It only improved the solubility twofold, but this was a different modification that we looked at as well. So for the orange series, the 44594 series, once again, here we have the HTS assays, the nanolook, the tech, and then the drug-like properties. We also looked at 3 different areas here. On this hit, we synthesized about 200 analogs. And shown here is, decent nanolook activity, mediocre tech activity. Metabolic stability is okay. It's not that great, but room for improvement. But the solubility and the permeability of this series was not acceptable at all. And then here we did get in vivo PK to confirm, because the ADME properties that we get, that's preliminary PK. You really need to get full PK to confirm the results that you're using as the ADME screening PK properties. And, once again here, we have very low exposure, low bioavailability. So in summary, for this series, the 45976 and the 45626 analogs, modest MLM stability, poor solubility, poor permeability, but we did have high nanolook tech triple combo activity. Not cytotoxic. We also did off-target profiling for one of these compounds. We did see some acetylcholinesterase activity at 6 micromolar, not very, very potent. We did look at other modifications, not in particular as all of the analogs of these two series, where we converted the amide linker to an oxidizole. We got really high nanolook response in there, but the solubility and the MLM also improved, but the permeability and the tech permeability was not great and the tech activity decreased. So in summary, we had 3 chemical hit series that were identified through a 500K screen. The hit to lead optimization of the hits was conducted through structural modifications at different sites of the hit compounds, which resulted in, several new analogs. And the identified new analogs significantly increased read-through when co-administered with low doses of aminoglycosides. So these enhancers may be a viable way to overcome potency issues associated with aminoglycosides, enabling rescue of protein function from nonsense alleles while minimizing toxicity. And of course, we need to do further SAR studies to improve the drug-like properties and identify efficacious analogs of proof of concept animal efficacy studies and further development. We also did look at some of these enhancers with ERF1 degraders as I was reviewing some of this data, and we did see an increase. And so, there's a lot of work to do in this area. But by God, we'll get there with a couple of preclinical candidates in hopefully the near future. With that, I would like to thank some of the work had begun at Southern Research, and all my colleagues there, all of our colleagues at the UAB CF Research Center, and the UAB Department of Biochemistry, which is now where I had moved to from Southern Research. And of course, our funding from the CFF, UAB, NIH with some of the early work, and Southern Research. And with that, I'd like to thank you for your attention.
Hi. A lot of data. So I was interested in the discrepancy that you found between nanoluciferase rescue and tech. And so do you counter screen against wild type nanoluciferase because of the terrible stories we all know about PTC 124 stabilizing luciferase, not having any effect on read-through?
Dave?
We don't routinely counter screen. We need to talk about that. We're at the
Right.
yes.
Okay. That's
Yeah.
fine. But do you so you don't counter screen, and then and then the same for just a wild type CFTR to counter screen there, because you don't know.
Yeah.
If you just get bigger activity, you don't really know the mechanism.
Right.
Yeah. I was just okay.
I was wondering if you did the screen on G418, did you try with G418, did you try some of these hits with other aminoglycosides?
Not as we were moving forward the program, we stuck with the G418. Yeah.
Okay.
But like if you administer them in vivo, you would pick like ELOX or something like that.
Yeah, I mean, there is a lot of data that we have with some of the key compounds with ELOX, but that's for a whole different presentation. There's a lot of data, and we are looking at a lot of different variations with ELOCs. Some of the work that came out, obviously, with the 99009 led us to then looking at it. I mean, there's a lot, but we have looked at selected compounds, and we have a ways to go, actually, with the different combinations, the triple combinations. We really need to make sure we're not missing anything with the activity that we're seeing and how we can get the biggest increase to get to our 20% wild type. Yeah. We're getting there, though.
Or more. 20% or more.
Yeah.
Hi, it's a great talk. So it's Heber from Foundation, and I have a question about the insolubility. So in the talk, I noticed the insolubility is below 1 micromolar. It's refer to the compound treatment or the compound stock solution. The insolubility of the compound is
we measure the solubility in SIF, stipulated intestinal fluid, at a 7.4 buffer.
Okay.
That's what the solubility number.
Thank you.
Okay, so if no further questions, thank you very much.
And last, we have Stephen Rowe, I guess, from UAB, who will be discussing the discovery of non-aminoglycoside small molecules that enhance the ERF3 degraders and induce translational read-through of CF nonsense mutations.
Great. Well, thanks for the moderators for inviting me to speak. Really happy to be with you today. I'm speaking on the capacity as my faculty member at the University of Alabama, Birmingham. But it's a really nice project that illustrates a collaboration between Corinne Augelli-Szafran and her previous work at Southern Research, now transitioning to UAB, David Bedwell's group, along with the CFFT laboratory. So it's a nice element of a collaboration and dual affiliation. And hopefully you'll see that we build on the work that we've heard about throughout today, including further information about Dr. Lisa Simpson's talk on ERF3 degrader series. These are my disclosures. It does include an unlicensed patent that's held by multiple investigators in this team on the use of these agents for translational read-through for genetic disease. So first introduction, premature termination codons affect about approximately 11% of individuals' CF, yet effective therapies for those individuals are limited. Previous studies have shown that the novel translation termination factor, the ERF3 degrader, SRI 47749, can induce translational read-through of CFTR nonsense mutations. Combining ribosomal modulators, such as aminoglycosides, with 47749 synergistically rescues those nonsense mutation read-through. However, aminoglycosides can be toxic at the doses required for effective read-through and have limited bioavailability, which limits their long-term clinical use. That includes ELOX-02, even though as a more potent and efficacious medication than G418, because of its limited bioavailability, really stands as a major limitation going forward. So the whole principal concept between our work that I'll share is try to develop an agent cocktail that could generate aminoglycoside independence. So to address this, we developed a high-throughput screen in the presence of the ERF3 degrader CC9009, and then applied medicinal chemistry optimization, such as what you heard about from Dr. Augelli-Szafran, to further optimize small molecules that can exhibit additive or synergistic effects with this ERF3 degrader 47749, and to do so in the absence of aminoglycosides. So first, a brief introduction. I think you're now familiar with this. By the end of this session, we're interested in translational read-through to circumvent the premature termination codons that generated the truncated CF protein. And the way that we've thought about this within our group is this is essentially equilibrium between translation termination and elongation, in part generating an equilibrium that can be influenced by the degradation of ERF1 and ERF3, which tips the balance towards translational elongation from read-through by generating a ribosomal pause, giving more time for things like aminoglycosides to modulate the ribosome affinity and generate the read-through. And we summarized some of that work was presented in a publication by our group, our combined groups in Nature Communications in 2021, where we put forward the ERF3 degrader, ERF1 degrader, SRI 41315. Now I'll talk about how we've extended this to other ERF3 degraders, such as we've already heard about in this session. Based on a medicinal chemistry approach similar to what you just heard about from Dr. Augelli-Szafran, we developed the ERF3 degrader 47749 and have gone on to show that it can enhance CFTR read-through, in this case, with both R1162X, as shown by the data here, as well as a variety of other premature termination codons, including G542X. This agent here shown in dose response by the transepithelial chloride conductance assay shows that it has a relatively high potency, including a reasonable bioactivity similar to the positive control G418 administered at EC50 concentration. You can see it also affects transepithelial resistance, but does so at higher concentrations of that chosen for further study, approximately 1 micromolar, which provides a 3 to 5-fold therapeutic index over its deleterious effects on transepithelial resistance. This 47749, a derivative of C9009, also augments CFTR. You can see that in the top line of the Western blot, achieving approximately 10 to 15% of CFTR expression by Western blot quantitation, compared to the positive control wild-type CFTR loaded at only 5% for comparison. You can also see that this agent is a potent ERF3 degrader, as shown by this Western blot, showing almost complete elimination of ERF3 at moderate and higher concentrations, less so at the lower potency concentrations that we've used for further assays. Here you can see quantitation of the overall CFTR protein. Of note, we've also monitored ERF1 as a potential mechanism, showed the distinction between the prior screen and discovery reported in Nature Communications, although there is some depletion of ERF1 at the highest concentrations. We attributed this to extensive ribosomal pausing, which has been shown in other complementary studies, including ribosomal sequencing. This generates within cellular mechanisms to degrade ERF1 as a secondary effect, and one that we're trying to avoid with these higher potency concentrations, lower concentrations of 47749. So the team devised a screen at the CFFT laboratory to try and move the project forward to generate aminoglycoside independent bioactivity. And they conceived this by using a nanolink reporter screen of a full-length CFTR with the nanolink label at the C terminus, and did so with the choice of the R1162X a mutation because of its relative affinity for translational read-through agents compared to the other common premature termination codons, as shown by a variety of molecules, including those that Corinne described from these previous series. So they screen compounds with the presence of bioactive amounts of C9009, looking for agents that might augment the ERF3 degradation, all with the concept that we could then ultimately apply it with SRI 47749 or other ERF3 degraders that may have clinical utility. So this describes the screen to hit a strategy that we took on after the screen was conducted at the CFFT laboratory. They conducted the screening in the reframe of a 65,000 compound library that included the reframe FDA approved hits along with a chemical diversity series library. From that, they reported 342 hits from this R1162X nanolink reporter. Then Corinne's team did the sort of medicinal chemistry approach that she applied and summarized in the previous talk. They ranked hits by the ELISA assay of the high throughput screen, then examined the assay interference by structural stabilities, looking for molecules that could be further derived to optimize efficacy and potency. Clustered those hits by structural similarity, considered PK and in vivo toxicity data, particularly given that many of those compounds came from approved drug library screen, for which there was known data. Then also did predicted cell cytotoxicity using an in-house AI model that was developed at Southern Research. And from all of that efforts, there were cherry-picked 20 different distinct series that we assessed for bioactivity. Today, I'm going to talk about the medicinal chemistry approach that we've had on one select series, noting that the other 19 series are also under current evaluation, and we have one that's a more fully developed series, and expect additional series to be developed from the other hits. We'll hopefully have an opportunity to share that next year. So this is an example of the bioactivity within this single series that all activated further function of SRI 47749 using the transepithelial chloride conductance assay for CFTR function. First, to orient you, here's the activity of 47749. Always uses a positive control for every compound that was nominated and developed through the med chemistry approach. This on the right side is the positive control G418 used at the EC50 concentration, and its reproducibility shown. Here you can see that over the course of the 6-month campaign, that multiple compounds were identified that augment the activity of 47749, ultimately residing on our current lead, SRI 50308. Among the 50 compounds in the series, it was highest bioactivity at 15 micromolar combined with the ERF3 degrader 47749. I'll show you a bit more of that data. This is the addition of SRI 50308 in the transepithelial chloride conductance assay administered with 47749. This is against the 16-8, the R1162X background in the 16-HBE gene-edited cell line. These are the current tracings shown at the top. Here in tan, you can see SRI 47749 generating approximately 5% of wild-type CFTR function. That can be augmented by about 10% when you add CFTR correctors. This is not unexpected. CFTR correctors have a variety of effects on nonsense mutation compounds depending on the mutation used. R1162X tends to be one of the more less responsive because of the underlying activity of the near cognate amino acids that tend to get inserted at the R1162X locus. If you were to do this sort of test with G542X, you'd see 50% bioactivity, and we've in fact seen that with this drug combination in G542X, although the level of read-through is less so. Here, then we added this new agent, SRI 50308. It has essentially no bioactivity as a read-through agent generating chloride current alone. However, when in combination with 47749, you can see it's augmenting bioactivity by about approximately 50%. Again, that can be further augmented when you add the correctors, Augelli-Szafran, tezacaftor. All of that is shown, including the tracings in orange and the CFTR inhibition with the CFTR NH172. Looking at the Western blot data from this same sort of experiment, here you can see CFTR being expressed in the presence of 47749, augmented by the addition of Augelli-Szafran, tezacaftor, and the potent ERF3 degradation with small effects on ERF1 as a secondary effect, just as I showed you in the previous slide. 50308, on contrast, has very little CFTR expression noted, consistent with the functional studies. But also no effect on ERF3 or ERF1, showing that it works through an ulterior mechanism. When those agents are used together, you can see the augmentation of the Western blot quantified in the graph below, as well as continued depletion of ERF3. ERF1 seems to be somewhat more affected. Again, we believe consistent with the secondary ERF1 depletion from enhanced ribosomal pausing. We essentially have then labeled this series of compounds ERF3 degrader enhancers, although it doesn't enhance the degradation of ERF3 per se, enhances the bioactivity and CFTR function by ERF3 depletion. Finally, we've looked at the ERF3 degrader in human intestinal organoid cells, grown in epithelial, high resistance transepithelial monolayers and assessed by first CFTR currents. Similar to the 16HBE R1162X cell line, here you can see in a donor homozygous for R1162X that 50308 compared to 47749 augments activity. You can see it augments activity by about 20% less though than we saw in the 16HBE cell line. But achieves a higher level of wild-type CFTR activity. I will note that the human intestinal organoids do amplify CFTR activity compared to primary human bronchoepithelial cells. So typically by about twofold. So our estimated bioactivity in human bronchoepithelial cells will be more about in the 10% range with this sort of combination. So in summary, the ERF3 degrader 47749 induces translational read-through but does not achieve clinically relevant thresholds of C activity as a monotherapy. The novel small molecule 50308 derived from this high-throughput screen conceived by the CFF team significantly augmented CFTR activity to approximately 8% of wild-type level when combined with SRI 47749. That is in the 16HB cell line. The 50308 did not influence ERF3 or ERF3-1 expression or induce translational read-through alone, indicating a unique mechanism of action that enhances the functional activity of ERF3 degraders. Adding CFTR correctors to 47749 or 50308 modestly augmented CFTR expression and function in R1162X. In human intestinal organoids, homozygous for R1162X, the combination increased CFTR-dependent function to approximately 20% of wild-type currents. SRI 50308 enhances translational read-through from ERF3 degraders like 47749 and can significantly restore CFTR function in primary organoid cells. The combination therapy involved, including these new ERF3 degrader enhancers for CFTR nonsense variants, looks to be promising, and we will continue the medicinal chemistry campaign on the other hit series identified in the assay. So I want to acknowledge that this has been an outcropping of a long history of collaboration, in part to develop the sorts of series of compounds that you've heard before, as well as the ERF1 and ERF3 compounds that we've published before. You can see the contributors, major contributors from my lab, along with David Bedwell's lab, the Southern Research UAB team under Dr. Augelli-Szafran, as well as the CFFT labs contributors. Happy to entertain questions. Thanks very much.
Gary?
Nice work, Steve. That's very interesting and very encouraging. And good to see you still doing research. That's great. That's a pretty profound effect on ERF3. So I'm wondering, what are you guys considering for searching for unwanted effects? Mass spec on these cell lines? I know the cells are viable, but I'm wondering if you're going to think moving forward to approval in humans at some point, what other kind of things can you do to look at?
No, it's a good question. I mean, especially when you're targeting ERF1 or ERF3 for that matter, although we did hear an interesting presentation that points out that these are still clinically approved compounds in the thalidomide series, but not without side effects. Yeah. First of all, we've got a target in the range of not complete reduction of ERF3. So you see the sort of concentrations that we're using in cells that try to approximate partial, not complete degradation of those molecules. And we are constantly looking for those with a therapeutic index as measured by the transepithelial resistance. Further than that, Corinne's team uses a traditional panel of toxicity assays to look for side effects. That's true for this series that has passed those, along with the 47749 series, which has actually gone on to animal studies, including those in both CF and other non-CF read-through diseases, and been tolerated in the range of 2 to 4 week type studies. So in summary, I think we're going to have to completely balance that. We think these series are tolerable in small animals, and we'll have to look to see how that goes.
Just still amazed you can knock out that component of translation. And it's not that toxic. I guess we're learning things about these systems that we just never envisioned ahead of time. So
Yeah, I think what we're doing is generating a niche for ribosomal alteration. If you go too far, I think that would be deleterious. We see that regularly as well. But I also think that there'll be a viable therapeutic index, especially in combination therapy where you're lowering the toxicity of any one of the component agents.
Hey, Steve, nice data. I realize that the reason for this is to actually get away from aminoglycosides. Read through, I think that's an admirable goal. But you kind of left us hanging there. Did you put G418 or ELX02 with these two?
Yeah. If you use aminoglycosides with those, you can see further additive of bioactivity with these agents.
Kind of because you left us at about 8% with the two compounds. What would that boost
Yeah,
it
it's
to?
about another 20 to 40% depending on the mutation you examine.
Okay.
With low concentrations of G418, which is the typical assessment that we will do.
And that's really good work. You gave a good rationale for testing it, I think, in 1162. But do you have any idea yet of how it works in the triumvirate of the three most common PTCs?
We've only looked at G542X, and we see a similar bioactivity in G542X expressing cell lines, with the exception of the corrector responsiveness, which I already mentioned. Heightened corrector response, bioactivity just on CFTR currents, about 70% of what you'd see in R1162X.
Yeah, this is a question I almost asked to the previous speaker. You know, it's so much work to do all that testing and further medicinal chemistry optimization. Like, at what point do you use the primary cells instead of 16 HBEGE to sort of prioritize which path you go down?
Yeah. Basically, we look for our cues as a team. So the number one, in my mind, is to push up the efficacy limit. The number one in the chemistry team's mind is to get to medicinal chemistry profile that can look like it can go forward. So we will sample within a series primary cells. Right now, we've been using the intestinal organoids because of the throughput associated with those. And then we've been moving some of the series back up to the CFFT lab to do the primary assays in a more blinded fashion to look for that. So that's our throughput. Right now, we're right in the middle of that progress with the 50308 compound, but it's part of what we did with the previous 41315 and 47749 series.
So building up a little bit on this from Scott, in your prioritization, do you actually also include like the diversity of PTC variants? Because this could also be a very strong point to push something forward.
Yeah, this has been a 5-year project, and so we have flirted a lot with the mutations and how broad to go versus how narrow to go. And of course, I've talked about the various different series, and you saw even further of some of the series that we partially shelved from Corinne and Corinne's work. Where we initiated our project is to look for using G542X as the putative mutation, and then once we had a lead series identified, extend to the other commons. And we've looked at W1282X and R1162X in that format. More recently, because of the relative propensity for read-through, as opposed to and greater CFTR function, even though you get less corrector potentiator responsiveness, we've transitioned to using R1162X. This is in part because if we really are seeking to get on the board with the next molecule that could generate aminoglycoside independence in patients, and we think R1162X would be the fastest path to that. But before any agent would go to humans, we would expect to do the typical and then in addition R553X, which we don't do in relatively high throughput, but have standing by once we're to declare a lead candidate.
Thank you.
Can I just ask one question following up on
we actually
Scott's?
have some time left. So if someone in the audience also wants to put questions also to the previous speakers, most welcome to do so.
Yeah, beautiful talk. And I'm just inspired to think about the type of cells that you've been acting on right now. So do you expect to see differences in the ciliated versus ionocytes in the secretory cells, will they all respond to the ERF3 inhibitors in the same extent that we get a fourfold increase in like CFTR levels? So what is your speculation
Yeah,
about
well, it's a
that?
great question. The first, and obviously extremely apropos on any kind of delivery target for genetic therapy where we've focused on that substantially. The answer here is I have no idea if that particular mechanism would vary substantially between the different cell types that you've mentioned. I would speculate that for the sort of mechanisms that we're looking at, ERF1, ERF3, or translational altering agents like aminoglycosides, that ought to be sustained across cell types because of the fundamental nature of translational machinery. We have not tried to do that by a cell type basis. We do use our primary human airway cells as a basis to making sure that that there's not such selectivity between cell types that you don't see CFTR vectoral currents.
Okay, thank you.
Hello, awesome talk. I was curious whether or not you had thought about maybe using the ASOs and decreasing the concentration of these drugs to decrease off-target, more maybe deleterious effects and increasing on-target effects.
ASOs for the ERF1 or ERF3 itself?
Blocking, as you were presenting yesterday, stalling the ribosome, allowing more time to find a tRNA.
Yeah, certainly we have played with ASOs, including those developed by Ionis that looked at ERF as one target and shown that they are bioactive in our hands. In fact, it was some of the basis for the original screen that resulted in the ERF1 degrader 41315. And you can read about that in the paper by Sharma et al. About that work. So we haven't since then returned back to ASOs because we feel like the small molecules that we have can hit that mechanism solidly. And now what we're looking to do is get combined therapy that hits the efficacy potential. We may be wrong, but part of the notion is if we have to use multi-agent therapy for nonsense mutations, we'd be much better use two small molecules than one inhaled ASO and one small molecule.
Okay. Yeah, just to add on to your question, so our lab has also explored this ASO approach that Andre presented yesterday a little bit, and so we have combined some of that with other read-through agents or the modulators, and there is definitely, again, like benefit. I mean, the idea of bringing down these doses further by adding it, and I mean, it's always a trade-off. How many modalities do you try to develop, right? And the beauty of Andre's ASOs is, of course, the specificity. At the same time, then you need to do many different ones. So we looking at all of those options, and ultimately we have to see what can be a safe therapy. That would be my response there.
Thank you.
Steve, can you speak a little bit, or Corinne, about the potency differences or the efficacy differences between CC9009 and this one? It seemed like you had quite a bit of activity with the molecule alone, just by itself, without the enhancer.
Yeah, we were getting to about 4% of CFTR activity, 4 to 5% in CFTR activity, about double that in human intestinal organoids by vectoral ion transport assays as a monotherapy. Its potency is worse than CC9009 in that CC9009 can have potencies in the mid nanomolar range, 50 nanomolar, I believe, is around the EC50 in our hands with that CFTR assay. Whereas we're looking at active concentrations in the 1 micromolar range. Having said that, the therapeutic index for the cell tox assays is by far favorable for 47749 over CC9009.
Being that there's a few minutes left, Scott's going to follow up on Scott. So,
okay.
And tell a little story.
He's Scott.
A little story. So at the beginning of the COVID pandemic, a good friend called me and he said, I take lisinopril, and there was some data about the interaction of lisinopril and immunity to COVID, and he was afraid, should I stop my lisinopril? So we put like mega dose of lisinopril on primary airway epithelial cells and did mRNA bulk seq. There wasn't one gene that changed. But when we put CC9 on primary airway epithelial cells, we got thousands of changes in mRNA. And if folks saw Emily's talk yesterday, when we did the whole proteome by mass spec, we saw 1,000 genes, 1,000 proteins change. So I wonder if we shouldn't be working these kind of assays like pharmacogenetics and, yeah, well, it's pharmacotox, right? I think that a pharma executive seeing thousands of genes and proteins changing after treatment with their drug would sort of freak out and kill the project. So I wonder if we shouldn't incorporate those kind of assays into these development pipelines. It's a rhetorical kind of question.
Yeah,
but
I mean, I think that in the ideal, go ahead, Martin.
I was going to say, if I can counter Scott just a little bit, I mean, yeah, we can use all the microscopes that we want and different assays and look at that. I mean, a lot of drugs have been safely developed by animal safety studies, right? So if you go in vivo and you don't see that these animals are ultimately suffering or so, then to me, that's still the preferred integrated readout compared to sort of worrying about every protein that I see moving left or right. I mean, that's...
Yeah, I mean, maybe I'll just, go ahead. You want to comment on this discussion?
Yeah, also with this is, we can measure all kinds of changes, right? But we don't know whether or not that change is physiologically relevant, right? Or is doing something that is outside of normal physiology. So I think, you just got to temper it. And yeah, if you don't see anything major in the mouse, then you kind of, that's been our standard. Then you move forward and you take your chances in the clinic, right? So
yeah,
until we can measure that level of, that threshold level of normalcy for everything, I don't know if we're ever going to know that answer until we take the chance.
I guess, one other thing to contemplate, because you're asking an important question, is in conceiving the idea of translational read-through. I mean, and to some extent, we're biting off a mechanism that may have the potential for untoward or effects because of its broad importance to human biology. What you're depending on is what's been shown repeatedly is the much greater potency for read-through agents to act on premature termination codons than native stop codons. So to some extent, I think as opposed to perhaps the advent of the original CFTR modulators where specificity on CFTR folding and not generalized folding was an important part of the portfolio, that may not apply as we consider it a read-through, but the proof will be in the pudding, and of course there'll be toxicity assays to follow to assure that.
I just want to follow up what Martin said, because we do look at these compounds and look for toxicity, mutagenicity, et cetera, when they start to look more promising. So I think those things would be caught earlier on than not in vivo if we go into animals. So, I think we do keep a broad spectrum of looking at different types of toxicities or mutagenicity changes. So hopefully we'll catch some of what we might be looking for.
So I would just like to point out that everybody that does these cell culture assays loves to see it completely disappear and to get the maximum effect. And that's not where we want to go in vivo, right? So these are essential gene products. And if we eliminate them, the cells are going to die and the organism is going to die. So we want to tweak 20, 30%, just enough to get some read through. And depleting ERF1 or ERF3 will probably sensitize the cells to read through, and these enhancers may further induce that. One of the effects of some of the enhancers is that they greatly increase the potency. So, for example, with ELX02, we can get by with 50-fold less to get the same read-through response. And so there are a lot of balancing things we're trying to do here besides Corinne's, metabolic stability and solubility and all that kind of stuff. So we are constantly arguing a little bit about, should we use this dose or that dose or both doses? And Corinne just says, I don't care, just do the same dose so we can compare them side by side, right? So this is complicated. That's the bottom line.
Okay, with that.
Any further comments? If not, thank you, everyone.